Hi, everyone. Welcome back to our chapter on sensory systems. Today, I really want to talk about the somatosensory system, so particularly pain and touch, and how that kind of comes into our body, how we sense it, how it goes through our central nervous system, and then ultimately respond. Part one of the video was about the auditory side of our senses, right? So hearing. And then we talked about that in class on Tuesday. This is part two. And then part three will be more on the chemical senses. So we'll talk through smell and taste, but we will do that in lab before we do a fun little lab experiment. And that will be on the 6th of October. So let's talk about the somatosensory system. This is the system that is responsible for touch as well as pain. And this is where our body is really connecting directly to the brain through these receptors that are in our skin and even in deeper tissues. And we can break down the system into three major parts. There's first what's called exteroceptive cutaneous system. And this is the one that most people think about when they hear the word touch. It's about sensing the outside world through our skin, right? And there are three kinds of stimulation that are happening here. There's mechanical stimulation, which is pressure or touch. So, like if I'm feeling my phone in my hand. There's thermal stimulation, which is temperature, and that's, knowing if your coffee is hot or if your soda is cold. And then we have the nociceptive stimuli, which is surface pain. So when you accidentally touch a hot stove or you prick your finger on a cactus, that is all exteroceptive, so outside of our body. There's also proprioceptive system, and this is our built-in GPS for our body position. So even when you close your eyes, you still know where your arms and legs are, and that's what is proprioception. That's what that's called. And so we don't have to rely on our visual system, but it keeps our body in balance. And then we have the interoceptive system, and this one is far more hidden, and that's because it monitors what's happening inside of our body. So think of hunger, think of thirst, or even feeling like our heart racing after we take a run. So the somatosensory system, I would say, isn't just about touching objects around you. It's really this three-layered system that helps you to interact with the outside world. It helps you to maintain balance and posture, and then monitor what's going on inside your body. And for purposes of this video and kind of this chapter, we're really going to be focusing on the exteroceptive cutaneous system. So kind of that outside body when we feel something that hits our skin. So we've looked at the overall system, and now I want to zoom in on specific types of receptors in the skin that actually make all of this possible. So there are four main types of receptors. We have free nerve endings. These are the simplest ones, right? They're going to pick up pain. They're going to pick up temperature. So if you touch something hot, these are the first to fire. Those are the receptors that are firing. You have the Pacinian corpuscles, corpuscles. And these are really deep in our skin. They're also very fast adapting. So these receptors are really great when it comes to detecting very sudden, very brief touches or vibrations. So when your phone buzzes in your pocket, that is a Pacinian corpuscle. We have Merkel receptors as well. Merkel receptors are slow adapting. They're more focused on steady pressure, and they help you really to detect textures, right? So if you are feeling little ridges on a coin, right? That is the Merkel receptor. And then you have the Ruffini corpuscles, and these are also slow adapting, but they really respond to stretch. So helping with grip and even like sustained touch. So you can think of, holding a cup and you're not dropping it, right? Now, I do want to show this, and this is not in your text, but I thought that this was really good. This is from another, I believe, textbook. But these are the receptors I just talked about. So the free nerve endings, the Merkel disc, the Ruffini, the Meissner's, and then the Pacinian. And we didn't go through Meissner's as much, but you can see here the structure, the sensation, and the location. And I really like how this puts it together. So you can see here these free nerve endings. They're actually unmyelinated dendrites of sensory neurons. And when we talk about unmyelinated and myelinated, if you remember, hypermyelinated makes it like very quick. Unmyelinated move a little bit slower. And so the sensation for those free nerve endings I mentioned are light touch, hot, cold, even pain that's coming in. And the location is actually around hair follicles and throughout the skin. So I thought that was pretty neat. And then Merkel's discs are expanded dendritic endings. Okay. And they have like 50 to 70 specialized cells. And its sensation is indented depth or sustained touch. And this is the base of the epidermis. The Ruffini are enlarged dendritic endings. They're open. It's a capsule that's elongated, and that's that skin stretch that I talked about. The Meissners are dendrites that are encapsulated in connective tissue, and that's what goes through kind of like the slow vibrations and the changes in texture. And then the Pacinian corpuscles are dendrites that are encapsulated by concentric lamellae of connective tissue structures. And this is the deep pressure, right? So very fast vibrations. And those are deep in our skin, right? Deep in the dermis. So here's just a cross section. It just shows, fat in the lowest layer that we can see here, and it's kind of marked. So I will go and I will show you my little laser pointer. So this yellow part right here is fat. And through this fat you're going to have arteries, you're going to have veins that are passing through, and that's going to be kind of the blue are the veins, and then the arteries are kind of here. They're those red ones. Now, you also have above those, you have different types of receptors. And so this is the slide that I showed you right here. This is where these are located in this cross section. So you have the Ruffy ending, which is a leaf-shaped structure on some of the nerve endings. And that is going to look like this right here. You have your Merkel's discs, which is very small bud-shaped structures. And so that you can see kind of right here, these little circles. You have your Pacinian corpuscles or corpuscles, and these are small, kind of onion shaped. These are these right here. And those are present in our nerve endings. And then the free nerve endings, you can see kind of right here. They're a little bit all over. They're smaller. They don't have any structures that are actually attached to them, and they touch the very upper layer of our skin. And then you have a sweat gland. And then the sweat gland looks like this right here. And that is kind of this convoluted structure. It's right above the fat layer. And then a tube arises from that. And then its end actually opens on the outer surface of the skin. And then, of course, you have hairs, right? And these are just the roots that are right here, right? The roots that are just above the fat layer, they're connected to nerves. Okay. So let's talk about one of two major pathways. So we talked, when we went through the vision chapter, we talked through the visual pathways. That's what your lab's on this week that's due on Sunday, is you explaining what the visual pathways are. We talked about the auditory pathways in class yesterday. We talked about the anterior and the posterior pathways. And now there are two major somatosensory pathways. And that is, one is the dorsal column medial lemniscus system. And this system is really primarily responsible for carrying information about touch, and even that proprioception that I mentioned at the beginning. And proprioception, in case you forgot, it's our brain's awareness of our body's position. So it's how you know where your hand is, even when your eyes are closed. And so here's kind of the step-by-step flow. So we have these receptors in the skin or the muscles that detect touch or body position, and they send signals into our spinal cord. So as you remember, everything that's coming into our body is going through our central nervous system. So it's coming through our central nervous system, through our spinal cord, up into our brain, right? We have neurons that are synapsing at certain parts. It's crossing through the thalamus, which is our relay station for nuclei, and then it's going to the appropriate part of the brain. And then we respond with a motor command. So, it's sending signals to the spinal cord, and then these signals actually travel up the spinal cord through what's called the dorsal column nuclei, and that is in the brainstem. Once it hits the brainstem, those signals actually cross over and decuss essentially. And from there, they are going to ascend through the medial lemniscus pathway. Now, from there, this information actually then travels to the ventral posterior nucleus of the thalamus, which again, I mentioned acts as a relay station. So everything that's coming in, remember, comes in through our central nervous system, so through our spinal cord, and then it may synapse, depending on what it is. It may synapse directly right when it hits the spinal cord. It may synapse right at the brainstem. It may go a little bit further, but eventually it will cross over, go to our thalamus, and then it will hit our appropriate pathway to the part of the brain. Now, finally, we have these axons that project from the thalamus to the somatosensory cortex, and that's where your brain is interpreting the sensation. So that's when our brain recognizes that, our arm is bent or we're holding a pen or a pencil. So if you think about it, this pathway is really why we can accurately type on our phone without looking at the keyboard. With us relying on touch and, proprioception constantly. The way we do that is through our dorsal column medial lemniscus system. Now, the second major somatosensory pathway is the anterolateral system. And unlike the dorsal column system, which carries information about touch, about how our body's positioned, the anterolateral system actually is specialized for pain and temperature sensations. So we have the touch here, and then we have pain and temperature going through this pathway. So here's how it works. When pain or temperature receptors are activated, their axons synapse immediately after entering the spinal cord. So in the last slide I mentioned, some may actually synapse right when they hit the spinal cord, some may actually go up to the brainstem. When it comes to pain and temperature, it is synapsing right as it hits that spinal cord. It's the first processing, and that process happens very early. Then the second order axons cross over, they decussate to the opposite side of the spinal cord. And then from there, those axons are traveling upward, but instead of just one path, they actually split into three separate tracks. And that's what you see on the screen here. So you have the spinal thalamic track, and this one is the main pain pathway. It is, again, projecting right to the thalamus. So it's going to the ventral posterior nucleus of the thalamus. Now, lesions here can actually cause a loss of sharp localized pain perception, so important to note. The next tract is the spino-reticular, and this route actually projects to the reticular formation in the brainstem, which we went through when we talked about brain structures. And then it goes deeper into that nuclei in the thalamus. And this pathway is important for more of the emotional side of pain, like the lingering side of pain. So chronic or dull pain, that emotion that comes with it. And when there's damage here, it can actually reduce the perception of chronic pain. Then you have the spinotectal tract, and this one actually projects to the mesencephalic tectum, and that's part of the midbrain. And it helps to really direct, our attention to painful stimuli. So again, removing your hand when you touch something hot. So together, right, these three tracks are really letting us know not just what hurts, but also where. And then they kind of recruit our attention so that we can respond very quickly. Now, this illustration just shows the steps in the pathway. So first step, the sensory neurons from the skin are actually entering the spinal cord through the dorsal root, and they move up through that dorsal column. And then from there in the hindbrain, which is this section right here, they synapse on the neurons in the dorsal column nuclei. And then the axons of these neurons actually move to the other side of the brain, and then they move up to this forebrain. And then three branches of the, trigeminal kind of nerve synapse happens, and then neurons from here cross over to the other side of the brain. So they're actually crossing over, and then they move up through that medial lemniscus and they reach the forebrain. And then in the forebrain, these neurons are synapsing on or in the ventral posterior nucleus in the thalamus. And then from there, they're projecting onto the somatosensory cortex. Okay. Now, this illustration just shows the steps in the anterolateral system. So in the spinal cord, the neurons are entering the dorsal root and they synapse right in the spinal cord. So this, you have it right here, and it is like it is already synapsing, right? Remember, pain and temperature immediately on the spinal cord. And the axons of these neurons then move to the other side of the spinal cord. So they hit, they go to the other side, and then they are ascending to the hindbrain right over here as the three tracks. So spinothalamic, the spinotectal, and the spinoreticulaire. And then in the hindbrain, the spinoreticulaire tract, as well as, three branches of the trigeminal nerve will synapse on the reticular formation. Okay. And then those branches fuse together, and along with these other two tracks, they actually ascend to the midbrain. In the midbrain, this spinotectal tract will synapse on what's called the tectum, and then the spinoreticular tract will come up through that reticular formation. Then you have the forebrain, and all three tracts are synapsing on neurons in that thalamus, right, in the nuclei, and that consists the ventral posterior, the intralaminar, the parafascicular nuclei, so on and so forth. So you don't need to know all of those terms, but just know they're synapsing, they're crossing, they're moving up, going through that thalamus and then going to that somatosensory cortex as well. Now, we followed kind of the sensory information up the spinal cord pathways. Now let's see where it lands on the brain, right? And specifically, we want to talk about the cortical areas of somatosensation. So when touch, when pain, when body position signals reach our thalamus, they get routed to three main cortical areas, and those three are right here. So the first one is the primary somatosensory cortex. This is also known as S1. And that is in the postcentral gyrus. This is the main hub. Okay. Then we have the secondary somatosensory cortex. And the somatosensory cortex is going to essentially process more of that complex information, so complex aspects of touch. And then you have the posterior parietal cortex, and that integrates all of our sensory input with movement, with spatial awareness. Now, I will say in the primary somatosensory cortex, things are organized in a very special way. They're organized somatotopically, and that means that different regions of the cortex actually map on to different parts of the body. And the mapping is called somatosensory homunculus. And it's like a distorted little body map, right? Where the hands, the lips, and the tongue take up much more space than, say, your back or your legs. And that's because those areas have more receptors and need finer processing. So, for example, if I were to draw a picture of a hand and I had my arm, I have these receptors here, I feel a sensation. It goes into my spinal cord. If it's pain, it's going to synapse right there. It's going to cross over. It's going to go up, up, up. It'll go into the thalamus. And then from there, it will actually, this hand part will probably go to my top right hemisphere up here, I believe. I think that's it. But it takes up more space. So it's going to be processed in a different part of our brain because there's so many more receptors here. Whereas maybe my back legs are not going to get processed in the same exact place. And so there are also, I will say, four parallel strips in our S1, so our primary somatosensory cortex, and each strip processing slightly different aspects of sensory input. So each one is a little bit different. So like texture versus pressure. What's interesting here is that when there's damage to the primary somatosensory cortex, it doesn't completely wipe out touch. And I mentioned this when we talked about the auditory piece of it. And when we were talking about hearing, that if there's damage to your auditory cortex, doesn't mean that you have total, hearing impairment. Same with this, right? It doesn't actually completely wipe out touch, but instead, people usually show some minor deficits in mostly tactile sensitivity. The reason why is because the brain is built with these parallel pathways. And so multiple routes are carrying the same type of information. So if one is damaged, others can still compensate for it. So there might be some deficits, but not total loss of touch. Now, I will say that this illustration just shows the primary and the secondary somatosensory cortex, and it's around the central fissure. So we looked at the auditory cortex in class yesterday, and this is that central fissure, and we had that in the temporal lobe, kind of right here. And then here we actually have the primary somatosensory cortex, and then this like little pink one is that secondary somatosensory cortex. And if you look, this is just literally kind of like the inside of it. And so you can see here, I mentioned like the body map and where certain things are taking up more room. As I mentioned, the hand, you can see here, like, look at where that's being processed. Like, this is all the hand. You have your face, you have kind of your like leg and your hip and your trunk and your neck kind of up here being processed. So it's pretty cool, actually, if you kind of spend some time looking at it. So once the somatosensory signals actually reach the cortex, the story doesn't stop at the primary somatosensory cortex. The brain actually will continue to process and integrate these signals in the association cortex, which we also talked about when we talked about the visual system, when we're talking about the auditory system. And so first signals get projected to the prefrontal association cortex, and it's heavily involved in planning and decision making and even like how we direct attention. So this is where sensory information really starts to influence higher level thinking, right? Like noticing when your phone is vibrating, right? Deciding whether or not we're going to answer that phone. They also go to the posterior parietal cortex, and that parietal cortex essentially is a key hub for integrating different sensory inputs, right? And what's, I think, especially fascinating here is the presence of bimodal neurons. And these neurons are responding both to, visual stimuli, to somatosensory, and then the receptive fields are very closely aligned. So for example, if I'm reaching out to grab a coffee mug, these neurons are going to help me match what I see with what I'm feeling, right? They're going to allow my visual and my tactile maps to work together so my hand knows exactly where to go without knocking this mug over. So you can think of the association cortex as really like the brain's collaboration space, right? Different senses come together and then they get linked to behavior and action. Now, I want to talk about what happens when the somatosensory system doesn't work as it should, specifically somatosensory agnosias. Now, agnosia means there's a loss of ability to recognize or interpret sensory information, right? Even though basic sensation is still intact. And so the first type we have is called stereognosia, and this is the inability to recognize objects by touch alone. So if you imagine, holding your keys in your pocket, right? You can feel the shape, you can feel the texture, you can feel the weight, but you cannot actually connect those sensations to the knowledge that those are the keys, right? Or those are my keys. Now, patients that have this can actually still feel pressure, they can feel temperature, but they can't recognize what they're touching. It's crazy. But then there's also asomatognosia. And I think we talked about this in maybe chapter 2, I think. But this is the inability to recognize parts of one's own body. So a person that has asomatognosia actually might look at their own arm and insist that it doesn't belong to them. And one specific form of this is contralateral neglect. And that's when someone fails to acknowledge, one entire side of space or their body. And it's usually opposite to where the brain damage occurred. So as an example, if there's damage on the right side of the brain, the person may ignore everything on the left side of the world. And so while the senses themselves are working, the brain's ability to really interpret and integrate that sensory information breaks down. And this shows us just how perception isn't just about detecting signals, but it's about the brain making sense of them. So I don't know if you have heard of this or seen this, but there is what's called the rubber hand illusion, and we could try it in lab. It works, or you can watch a lot of YouTube videos on it. But this is a common induction method. And the participant's hand, what happens is they have like a board or something, and they have their real hand kind of on the outside of it, so they can't see it. And then there's a rubber hand that's in front of them. And then what the experimenter does is they're actually maybe taking something and they are maybe stroking both the rubber hand and their real hand at the same time. And it's cool because what ends up happening is they almost mistake the rubber hand for their own hand, and it's fascinating. And it happens to all of us actually, probably more frequently than we would think. But there's lots of cool videos on that. You can see that happening. Now, okay, although most people think that pain is bad, it is actually very adaptive. And so the perception of pain is pretty fascinating. I will also say that there are cases of what's called congenital pain insensitivity. And those are people that literally do not feel pain. And I believe there's a story in our text about a case of a woman that like could not feel pain at all. And eventually she passed away. Like I think she was in her 20s, because she ended up getting infections and she couldn't feel pain. And so that does happen as well. Um, so the cortical representation of pain is actually very complex. So you have your S1 and your S2, so your primary somatosensory cortex and your secondary somatosensory cortex, and both are activated by painful stimuli. Okay, but removal of both areas doesn't actually change the perception of pain. So we talked about some of the biopsychological methods, research methods, and we talked about the PET scan, right? The PET. And researchers have actually found that the anterior cingulate cortex has been consistently implicated in cortical perception of pain. Now, it does appear that this activation is more related to emotional responses to pain rather than the perception of pain. Also in 1965, I believe, Melzack and Wall were impressed by this ability of emotional and cognitive factors to block pain. And so they conducted that there must be some kind of circuit that descends from the forebrain that can actually block incoming pain signals. This is called gate control theory. And the first direct evidence of such a descending circuit really came from the discovery that stimulation of the periaqueductal gray, so PAG, produced analgesia in rats. And surgery could be performed under this stimulation. Next, it was discovered that there were receptors for opiate analgesics. And I think that like morphine is one of them, and several other sites. And this suggested that the body actually produces its own opiates. And they're called endogenous opiates, or we know them as endorphins. And several were subsequently kind of isolated. And then there are several lines of evidence that led Bosbaum and Fields to propose a descending I don't know how to say it, analgesia circuit. And the circuit goes from this PAG that we talked about to the raphe nucleus and then descends via those dorsal columns into the dorsal horns of the spinal cord. And the absence of pain has been linked, and this is important, I think, for you to know and take away from this slide, but the absence of pain has actually been linked with a gene that influences the synthesis of sodium ion channels. And this discovery, again, being that it's kind of in its infancy, right? This discovery can actually lead to a creation of a new generation of like pain being adaptive, right? Or analgesics. Now, I do want to go through this last slide here and talk about neuropathic pain. And neuropathic pain is really severe and chronic pain, but it's in the absence of a recognizable pain stimulus. And oftentimes it develops after an injury. And it is thought that our glial cells are triggering hyperactivity and neural pain pathways. So we have pain that's adaptive, where there are some people, and the gene causes this, that don't feel pain at all, ever, which is dangerous. And then we have people that have severe and chronic pain, but we don't really necessarily know what causes it, right? There's not a stimulus. I'm not taking a needle and poking myself and knowing that the pain is coming from that. I'm just feeling pain. So that is what is called neuropathic pain. I will stop here. That is part 2 of chapter 7. So I strongly suggest you read your text. And when we meet again in lab on the 6th, we will talk about the chemical senses and go through smell and taste. Thank you, everyone. Have a great week.